I'm Mary Beth Gilliam, and this is the Outperform Cancer podcast, where we discuss actionable anti-cancer strategies found in peer-reviewed scientific research. If you've been enjoying this podcast and would like to support it, the best way to do that is to take a moment to follow the podcast, click like, add a comment, or best of all, share it with a friend. You can also visit outperformcancer.com and sign up for our newsletter. Today, we're talking to Dr. Abby Overacre. Dr. Overacre has a PhD in microbiology and immunology from the University of Pittsburgh. The Overacre lab focuses on the intersection of the microbiome, immune system, and cancer, especially as it relates to immunotherapy. Today, we're translating that information and all that hard work she's done into something really tangible and easy that you can do for yourself to help improve your outcomes. What if something as small as the sweetener in your coffee could influence whether your cancer treatment is going to work? Wow. This is a big finding. Dr. Overacre and her team at the University of Pittsburgh Medical Center recently published a groundbreaking study in cancer discovery showing that sucralose, the artificial sweetener found in Splenda and many other sugar-free products, can actually block the effectiveness of immunotherapy by disrupting the gut microbiome. In both mice and patients, higher sucralose intake was linked to poorer responses in treatment. And we're not talking about doses of sucralose that are out of the norm, something that no one would ever eat. We're talking about doses as little as 3 packets of sugar, having a massive impact on whether immunotherapy drugs work.
Some tumors were very big and very aggressive, and some were not. And when you see things like that, that's so black and white, it becomes hard not to make a change.
This is a critical discussion. And we're going to dive into this fascinating research with Dr. Overacre, exploring what it means for cancer patients, how the microbiome connects what you eat to your immune system, and whether everyday choices about sweetness could really have life or death consequences. Joining me in welcoming Dr. Abby Overacre. Dr. Abby, thank you so much for joining us. We're really happy you're here today.
Yes, thank you so much for having me. I'm excited to be here.
Well, I was blown away by the article that I read recently that was titled, "This artificial sweetener could make cancer treatment less effective." And I knew that many other cancer patients would want to know what you had to say. So right off the bat, can you tell us what research this is referring to and what was your big finding?
Sure, yeah. So this was a paper that was recently published in Cancer Discovery by my group. And it was a bit shocking to us, actually, the findings. So what we found is that artificial sweetener use, so these are things that come in like the yellow and pink packets that you put in your coffee, that these are actually very detrimental for immunotherapy in patients with cancer. And what was really interesting about this, so first of all, the surprising part actually is that when you put this head to head against just table sugar, within moderation, of course, but when you put those two things head to head, artificial sweeteners are actually much worse compared to just table sugar. So that was quite surprising.
So when you say much worse, tell me what you mean.
Yes. So essentially we looked at this through preclinical models where we could directly test, in these models if there was consumption of an artificial sweetener like sucralose versus just putting sugar in a water bottle, right? That there were 2 parts of this. So one, the tumor seemed to grow a little bit faster in those that were consuming artificial sweeteners. And secondary to that, and perhaps more strikingly, the immunotherapy response was completely blunted if they were consuming artificial sweeteners. Whereas if they were consuming table sugar, we did not see that same impact. So the immunotherapy could still work.
Wow. Okay. So I'm going to rewind that for just a minute because I want to make sure that everyone gets this point. So, what you're
Yeah,
saying is that you saw two things that were happening that are really important for a cancer patient to know. Number
right.
one is that you saw the tumors increase or start to grow. So you saw
Yeah,
promotion of tumor growth,
yeah,
and that was with artificial sugars versus regular sugar. So
correct,
increased tumor growth. The second thing was that the immunotherapy that they were using was not working as well.
correct, correct. And so this was all shown in preclinical models, and the beauty of that is that you can determine what's causing something, right? Because you're in control of the experiment. But, of course, those can only tell you so much, right? We have to learn from patients who are willing to, provide information or provide tissue samples. And so when we went and looked at both, in this case, it was melanoma patients and non small cell lung cancer patients, we were able to go back and ask, a very extensive diet history questionnaire. We're very thankful for patients actually taking the time to fill those out. And that was really where we could see this striking difference in how well patients, how much benefit immunotherapy was providing to patients, right? And so essentially in both cases, and these were two different immunotherapy regimens that they were on and two different cancer types, patients who self-reported as consuming these kind of artificial sugars, artificial sweeteners, had a significantly worse overall prognosis compared to those that were either consuming none or very little.
I want to clarify worse prognosis. Did
Yes.
that mean worse overall survival?
Yes. So we looked at a couple of different things. So we looked at, because there's all these different terms that are used, right? So across the board, we looked at overall response rates to immunotherapy, those were decreased. And then progression free survival was also decreased. So those are the two main things that we've looked at. We also will have overall survival for these patients. We already do for some of them, but lots of these things are still in the works.
Okay, so no overall survival data, but progression free survival means that you're tracking how long someone goes without their cancer progressing. So if they were
Correct.
in remission, how long did it take for that to come back? Or if the cancer exists, how long for it to start growing, actively growing? So you did see that there was a shorter time that patients got until they saw their cancer grow again. So we
Exactly.
see, and usually my understanding is that progression free survival often goes hand in hand with overall survival. So if your
Yeah.
cancer is
Yeah.
starting to grow faster or
Of course.
sooner,
Mhm.
then a lot of times you have worse overall survival. Is that correct?
Yeah, absolutely. And, I think that one of the things that we think about all the time is that immunotherapy, and we say this all the time around clinicians and other researchers, is that immunotherapy has been paradigm shifting for treating cancer patients across different types of cancers. And this was something that came about when I was still in training, that it really took hold. And, there was a Nobel Prize awarded for discovery of these things called checkpoint inhibitors, which is the main immunotherapy people use now. It's been very successful. And so this was a huge win for immunologists, for cancer research, of course, for patients. Um, but unfortunately, the majority of patients still don't benefit or still don't see their tumors shrink as a result of immunotherapy. And so, when it works, tumors can completely go away and people are truly cured, which is always the term that we were afraid to say. But we still have a lot of work to do. There's still a lot of patients who don't get a benefit from it.
Hey, can I stop you there for a
Yes,
second? Because I think that sometimes patients can get confused by that. So
sure.
they feel like, gee, a drug should work or it won't. If
Yeah,
everybody takes an aspirin, everybody
yeah,
kind of
yeah,
has pain go away. So
yeah,
can you
yeah.
help us understand um, if there is a group of patients, of cancer patients, and they're taking these immunotherapy drugs?
Mhm.
Yeah, about how many of them respond versus don't respond. Do we know that information?
Yes, we do. And it is a really good point, right? Because, yeah, I mean, you expect to take an aspirin or something and it should do its job, right? And so yeah, there's a couple of points, I think. So in terms of percentage of response rate, so this varies widely depending on the type of cancer. Melanoma has been kind of the poster child of immunotherapy and especially checkpoint inhibitors. Um, and so again, it depends on the treatment and the type of cancer, but we're looking at, on average, it's anywhere from, it's often somewhere between 20 and 40%. And those numbers keep going up, which
Who
is good.
do
So 20 to
respond?
40% respond. Now,
Okay.
it sounds low, but so the way that these actually started, and patients will probably know this better than we do, right, is that these all had to start as a clinical trial, right? Various clinical trials. And so this requires, of course, patients who have done standard of care, whether that's chemotherapy, surgery, radiation, a combination, and they essentially have to fail everything to be able to go on one of these new clinical trials. And there's good reasons for that, right? So immunotherapy had to be kind of this last resort thing. And what they found, which was really exciting, is that even with those patients who often have like the hardest to treat tumors, that immunotherapy was working for 20 to 40% of them. And that's actually huge when you think about it. And so what happened from there, thankfully, and they're still going through, they're still trying to figure out what type of immunotherapy is best for each type of cancer. Now combination is really important. So combining different types of immunotherapy. But what they've been able to do in cancers like melanoma, now others are following as well, is actually move up when you can get immunotherapy, where perhaps you could get this, at the very beginning, right? Rather than needing to undergo all these other things to get there. So I think that's really important. And the other, I think there's two other things. So one is that the percent of people we're seeing respond keeps increasing. And this is in part by when we're giving them immunotherapy, but also, the thing that I talked about, which is combining different therapies. So
Mhm.
there's been this explosion of, kind of identification of new treatments that we can combine with immunotherapy to make it work better. And so I think that's actually helped a lot more patients overall.
Interesting. So, but I think, you make a very important point, which is it is not a universal response. Some
Mhm,
patients respond, some don't.
mhm.
So that's why we really need to be looking at what is happening with these responsive patients and
Yeah.
can we get others to respond as well?
Yes. So and this is the thing that we all spend so much time thinking about. And this is kind of what started this project, actually, is that we now know from some of my collaborators at the Hillman Cancer Center, but also elsewhere, that the gut microbiome is critically important for patients to respond to therapy. Um, and to kind of break that down a little bit further, what we know now, and again, it started with melanoma, but now it's spread and followed suit with lots of other cancer types, is that cancer patients, if you broke down, patients by those that, did respond to immunotherapy versus did not, you could actually, pull those patients out just by the fact that, by different gut microbes that they had. So responder patients had a high level of certain gut microbiota or certain bacteria that live in your gut, and non-responders had different ones. And so that was huge, right? That opened up a whole new area that we could start to target. But then the really cool stuff came when people started saying, okay, well, how do we change that then? Right?
Yeah.
This is something that you can change. And so the earliest look at that really had to do with what's called a fecal microbiome transfer, or an FMT. And so what these did is they took patients who did not respond to immunotherapy and they reconstituted, or they replaced their gut microbiome with a gut microbiome from patients who did respond. Okay, so everything else is the same, but they just swapped out their gut microbiota. Um, and when they did that, then half of those people who didn't respond then responded to immunotherapy. And
Wow.
that is huge jump, right? I mean, that's just, to put it very plainly, like we always, make kind of poop jokes, but it's essentially like you're replacing that part of your intestinal system, right? And you do
Right.
nothing else, and now immunotherapy works.
Okay, I know there's going to be people out there that are wondering,
Yes.
how exactly
Yes.
do you do that? That sounds so gross. I
It
don't
does
want
get so
to do that.
gross. Yeah, so this is always a question. So there's two different ways that this can happen. And so the first way, which is how it first started, was through like it's a scope, essentially, like think about a colonoscopy, something like that. Um, and so that was how the original studies were done. However, now what most trials and most groups are doing is they actually just put them into like a capsule, into a pill. And so most patients, they end up taking, it's quite a few pills, maybe 5 or 10 pills. And I actually asked one of the clinicians who does this, and I said, how do patients handle this? And he kind of looked at me and he was like, these patients do so many harder things. He was like, they have no problem, right? They take them and they really don't have side effects to that. So yeah, so now it's just another pill that you take. That's it.
And that's it.
Mhm.
So these patients, we learned through research that these patients who had a reseeded gut microbiome, and I am going to just chime in that
Yeah.
I've read that if you have a fecal transplant, so if you have your gut microbiome reseeded with
Mhm.
healthy bacteria, that if you continue to eat a diet that
Mhm.
does not have very much fiber,
Mhm.
is primarily processed foods, has a lot of artificial sweeteners in it.
Mhm.
If you continue that diet, it won't matter that you've
Mhm.
had a fecal transplant, that the fecal transplant only helps if you actually change your diet and feed that good bacteria with good fiber, etc. Is that true?
So I think there is a yes. I think that is true. So I think that a fecal microbiome transplant it's a huge shift to your gut microbiome often, right? And so that will give you a benefit for a while because it takes time to have drastic changes from diet. However, what you said is very true, which is that there a lot of what we eat every day, it fuels us, but it also fuels all of your microbes. So it turns out that there are certain things that we can't even digest without the help of the gut microbiome. So certain fibers, when you're eating all of your high fiber foods, those are actually your gut bacteria that break that down for us and allow us to use those nutrients. And so, kind of the best way to think about it, I think, is that, for people, there's carnivores, there's vegetarians, there's vegans, right? And they all eat different things. So if you imagine that, but on a much larger scale, that's kind of like your gut microbiome. So sugars will feed certain types of bugs, certain types of bacteria that live in your gut. Fiber will feed different types of bacteria. And perhaps unsurprisingly, lots of the bacteria that we know that can be beneficial do like things like high fiber, and things that like a lot of sugar often are not good for a healthy gut microbiome. So yes, absolutely. So what you eat does matter. And it's the quickest and the easiest way for you to change your gut microbiome in a good way or in a bad way.
And how long does it take, though? Let's say that someone's listening to this now
Mhm.
and they say, wow, I didn't know. Now I know, and I'm going to make some changes.
Mhm.
How long after they decide to eat kale,
Mhm.
how
Yeah.
long after they decide to actually change their diet and have whole food, plant based
Mhm.
diets,
Mhm.
does it take to impact your gut microbiome?
Yeah, so this it's a complicated answer, but I will kind of say the things that we do know. So
Okay.
I think that the positive side of this is that as soon as you there are some direct impacts, right? So as soon as you start eating more salad, eating more kale, eating more something like that that has higher fiber and higher nutritional value, you are directly feeding bacteria that same day that are good for you, right? And then for like the longer term shifts, there's been a little bit of work done on this. I think there's some really good groups out at Stanford that look at this quite frequently in terms of especially plant-based diets versus meat containing diets. We think about this some with our collaborators with ultra processed foods or different sugars, right? And so a lot of those things you can see pretty strong changes within a couple weeks. But it's a it's a complicated answer because you know just like people, maybe some bugs are a little more stubborn than others, so it can be
Okay.
hard. Some take longer to shift or to change than others. So
Okay.
I think the positive of this is that your diet will have automatic impacts, and we probably all have noticed that, right? You eat healthy for a day, you do feel better, right? And part of that is gut microbiome driven. But I think it's just, it's about if you're going to change your diet, changing it and then being consistent, right? So you can get those long term benefits as well.
Awesome. Awesome. So we've shifted our gut microbiome. We've started to
Yeah,
make some good decisions. But I want to understand and come back to the immunotherapy and the specific drugs. You were talking
yeah,
about melanoma and non-small cell lung. Can you tell me what exact immunotherapy drugs were looked at so
yeah.
that people listening could say, maybe that's me?
Right. So the common thread with this across these different cohorts of patients is anti PD-1. And so anti PD-1 it's a checkpoint inhibitor, and probably the most it's certainly the most well-known immunotherapy right now, and probably the most used, I would say, if I were to guess. So other names that people might know this as could be like nivolumab, but also if you see commercials for like Keytruda, those sorts of things. Those are the ones that we're talking about.
Okay.
And I think what added to this is that it wasn't, all of the patients that we looked at did receive anti PD-1, but some also received other drugs as well, including like a TLR9 agonist. And these are all trials that were run by my very close collaborator, Dr. Diwakar Davar. So he's a medical oncologist who treats melanoma at UPMC. And so he was doing a few different trials. And so yes, the commonality is that they all got anti PD-1, but then they had other things as well, added in certain cases. And I think that it makes it perhaps a stronger finding because it might be more universal. We are now very interested in looking at this in different classes of immunotherapy. So
Mhm.
these are again all in that kind of checkpoint inhibitor category. But patients also, of course, get things like CAR T cell therapy or TIL therapy. So there's a variety of different things that we could hopefully look at and see if it impacts those as well.
Okay, so all of those therapies. So essentially, while the anti PDL1 immunotherapy drugs are the ones that are predominant and the ones that have been used in a lot of this testing, what you're saying is that the class of drugs,
Yeah.
immunotherapy, are something that you think have a relationship with the gut microbiome in terms of
Yes.
its, maximizing its effectiveness.
I think that yes, and I think a lot of yes. So I think part of the statement is based on what we already know from doing some not my group but the other clinical trials that have come out, very broadly looking at the gut microbiome and different immunotherapy modalities. And then some of it has been, we're just starting to get into it, right? And trying to say in more detail, not just saying, okay, we need a healthy gut microbiome, right? Which we know, but saying, okay, what can patients do or what can the clinical teams do to help get the biggest benefit for their patient?
That is a great segue back to this article where we started, which
Yeah.
was this research that you did identified something that is a key to unlocking the relationship between the immunotherapy and the microbiome. Can you talk to us about what that is and what you saw?
Yes. So the thing that we often talk about in the lab and what really pushed us in this direction is, it's great to identify a problem, to identify a reason why patients aren't able to respond to immunotherapy. But it's much better if you can come up with a way to fix it, right? Um, and so we always approach things in this way, and on top of that, having great clinical collaborators to kind of keep you grounded as a researcher to say, what is feasible for patients and
Mhm.
what's not, and how do we translate this for patient benefit, right? So with all of that in mind, we did a lot of digging on how things like sucralose and these other artificial sweeteners were impacting the anti-tumor immune response. an immunotherapy response. And so we noticed a few things. So we first noticed that the gut microbiome was heavily impacted. And this was associated with changes in microbial diversity, which is just how many different types of bugs are there. And typically, if you think about it in a general sense, a more diverse microbiome is typically better. And then on top of that, we also did further digging on these other things. So at a very, very small scale, what metabolites are being produced. And so metabolites are different. You can kind of think of it as fuel for cells or food for cells or bacteria. And so what's interesting is that your gut microbiome doesn't just take in and eat food and help you digest it. It will also kick out or excrete things that we can then use that are very beneficial. And so that's kind of the relationship between, food, the bugs, and us. And so what we noticed is that there was a significant loss in arginine, which is an amino acid that's very important for a lot of things, but it's extremely important for immune cell health and specifically T cells. And when we think about, T cells and cancer, T cells are the ones that often go in and actually do the killing of the cancer cell. So we need these cells to be primed, ready to go, full of energy, to where they can go in and attack. And without arginine, they can't do that. They kind of view arginine as a fuel source. And so what we found is that by just adding this seemingly harmless sweetener, the gut microbiome was shifted and there was less arginine in the gut, but also in circulation and also in the tumor itself. So this really was a systemic drain on arginine. And so from there, we started thinking about, we of course could try shifting the diet back to a healthier diet. But the other thing we wanted to think about is can we kind of meet patients where they are and, try to give them a boost or a benefit by directly giving arginine back. And so that's what we did. So we ran tests preclinically and we boosted arginine in two ways. So there's two ways you can think about doing this. So one is you can just give arginine directly, orally. The other one that we actually think works better is citrulline, which is another amino acid. But what's interesting is that citrulline, as it gets broken down in the liver, it actually gets converted to arginine. So you can think about this kind of as it's like a precursor almost to arginine. But the important thing here for people who actually take it is that if you compare arginine levels, let's say in circulation of a healthy person, right? And you compare someone who's taking an arginine supplement or taking a citrulline supplement, those taking a citrulline supplement will have higher arginine levels in circulation compared to those that just take arginine.
Okay.
And that has to do with just how your body breaks things down. And so, yes, go ahead.
Sorry, I just wanted to ask a little bit about dosing. So, how much are we talking here? Like we're talking mice and then humans. So, what was the dose that the mice got? And then how would that equate to what a human would have to do in order to have a similar response?
Right. Yeah. So this is always tricky because, of course, mice are much smaller than humans and their metabolic rate or their metabolism for their body is very different than ours also. And so the mice, what we did in both cases, actually, because this also applies to sucralose consumption, is we tried to take like a normal, what you can consider a normal human consumption rate, right? So, and I can break it down for both of these. So for sucralose, that was basically 3, if you put in yellow packets into your coffee, it's equivalent to having 3 yellow packets a day, which is actually not that much for some people, right? And so essentially what we do is there's a mathematical equation to take that or the arginine level, and the arginine level was just taking like a regular supplement level. So like 2 supplements a day, right? That you would just get at the store. And so essentially we have a mathematical equation to take a human amount for a regular average human and shrink that down both by size and by metabolic rate. And that's what we gave for mice. And so this isn't an astronomically high amount of arginine. This is just you go to the store, you buy the supplement. And we haven't done the clinical trials for this in patients yet. This is something that we're very interested in doing. Just to say, if you have patients that already have this kind of type of diet where maybe they're a high artificial sweetener consumer, certainly changing the diet, but also could we try these different supplements. And for something like that, I think we would have to look at different doses just to make sure that we get to the optimal level in circulation. But it's not a crazy high amount.
Okay. Okay. I had read in another study that a different type of gut microbiome called Akkermansia,
Yes.
when that is absent, that patients could also have less of a result from immunotherapy. So, in other words, if you were deficient in Akkermansia, perhaps you weren't getting as much impact from that immunotherapy drug as you should or as you could. But I also read that if you supplement with Akkermansia and you already have a healthy level of Akkermansia, that you can actually hurt yourself, and then you can have too much, and then that would also hurt your results with the immunotherapy drugs. So do we know anything about arginine? Like if someone said, oh, wow, well, I'm going to, eat my Splenda and I'm going to take some arginine. Do we need to know, if they have healthy arginine levels or not before they start supplementing?
Sure. Yeah. So, I mean, so the first of all, the study you brought up is extremely important, right? And so I think that that's worthwhile taking a second to talk about because Akkermansia is this drug. It is a bacteria that has gotten a lot of attention for being a beneficial bug, and it is. But what you said is exactly correct, which is there is kind of this like Goldilocks idea here where if you don't have it, we know really without a shadow of the doubt for many different diseases, it is not good. It is a very important beneficial bacteria to have in your gut. However, as you mentioned, if you get too much of it, right, then this is actually a huge problem as well. And people are still trying to understand, what it is about that. Like, why is it so important? I think that, we can certainly speculate again that having a more even, diverse group of bacteria is better than having any one outgrow. But that one is a very interesting example. And it's something that we have actually seen across projects as well, where too little is bad, but too much is also bad. So for things like arginine supplementation, so I think, it's early days in terms of trials and things like that. So, I think taking a regular amount of, an arginine or a citrulline supplement, it's been shown time and time again to be well tolerated, very safe. So I think that would be fine. I think, if there's somebody listening who says, okay, I'm going to keep consuming all these artificial sweeteners, but then take a boatload of citrulline, I think, that would be a time where maybe, getting a better sense of, talking with your physician, getting a better sense of where your blood levels are at, what does your gut microbiome look like? Because, I think the other thing to note is citrulline or arginine supplementation certainly provided a benefit in our preclinical models, but humans, of course, are way more complicated than mice, and we have a much more diverse microbiome. And so I think, we'll see what we find with the arginine supplementation. I hope that it kind of overrides any negative effects of artificial sweeteners, but of course, I would still, if somebody was asking me for my opinion, I would still say, well, let's try and cut those out too, right? Yeah. It's a decision that I actually have made after doing these studies, right? I was an average consumer of, 3 packets a day or loved my diet sodas. And I did shift away from doing that. So,
well, that's always super meaningful. I like to ask doctors when I see them or they're making recommendations, what would you do for your daughter or your mother or your sister, or yourself, as you were saying, like that you've made that change for yourself says a lot about about your confidence in your findings.
Yes, I think it's, whenever you conduct the experiments yourself and you see the results firsthand, it's almost like you don't have a choice. You just see how obvious it is and how different it was. And whenever you're looking at something, it doesn't require measurements, right? In these cases to see the difference, it was just some tumors were very big and very aggressive and some were not. And when you see things like that, that's so black and white, it becomes hard not to make a change, I think. Yeah.
Wow. Yeah. That I hope all cancer patients out there hear that loud and clear, because this is something that they have control over. It's something that they can do. And just to reiterate, we can have a separate discussion about whether sugar is a good idea for cancer or
Yeah,
not.
sure.
But even in this narrow field, what we're
Mhm.
saying is if you're going to have something sweet, you're better off having sugar, right?
Mhm.
Regular old table sugar than you are an artificial sweetener, especially sucralose.
Yes.
And many people will say, well, gee, what's sucralose in? I don't know. Is in many things.
It's in so many things. So, and I learned about this because we're doing these studies and we're looking at a lot of different artificial sweeteners, and some of them are more well known. Everyone kind of knows aspartame is in certain diet sodas and things. But sucralose, it looks a lot like the word sucrose, which is table sugar. So that I think throws people off a little bit. But what I have found is it is in certain diet sodas. It's in a lot of energy drinks that are very popular. It's in certain protein bars that we think of as being like really healthy. So the thing that I have noticed is that anything I would be checking very closely. Anytime anything says sugar free, essentially, unless it actively says it's stevia, because that's a natural product, and so they like to advertise that. But essentially, if you're looking at a food or drink that says sugar free or diet, so like lots of times sugar free syrups at coffee shops, same thing. If you actually just look at the back, it probably has sucralose in it. So
I
it's one of those, and it tastes just like sugar, you know? Um, and so I think that's why you don't always know. If you eat something with stevia in it, you probably know. Or if you eat something with monk fruit in it, you probably know. Even aspartame has a very specific taste.
right.
People love it, but it has a specific taste. Um, sucralose tastes like sugar. It's from what I have seen at grocery stores and checking things. This is probably, I think it's probably the most commonly used artificial sweetener in terms of breadth across different things.
Yeah, and I'll just add because I know that your research doesn't name products, which I
Yeah,
understand from a research community perspective, that's not
yeah,
what you want to get into. But
right.
since I don't have to worry about
Yeah,
that,
yeah,
I'm going to just let everyone know that it is in Splenda, is one of the products that it is in. Now, Splenda does offer a stevia version, just
yeah.
so people
But
know.
most of those still have sucralose in them, I think.
Possibly, yeah.
I would just check. Yeah, always check the labels. Always
Always
my
check the labels. And then yeah, I found it in lots and lots of products. Lots of
Mhm,
products. I was talking to my son about this,
mhm.
and he, much to my dismay, had a Monster energy drink and he's like, "Oh, it's in this." And I
Yes,
said,
definitely in
"Well,
those.
I was
Yeah,
like, okay, it's out." He's
yes,
like, "All right, I'll go back to my black cup of coffee."
yeah.
But yeah, it is in a lot of products. So everyone should just look very carefully at that. And then, consider, I just also want to know that we're not making any recommendations for anyone to take arginine. That is something that you will have to make a decision for yourself as a patient and with your doctor. So please talk to your doctor if you have questions about this. I would suggest print the research. This research was in Cancer Discovery, and you can go and look it up. There's also summary articles that are available at the University of Pittsburgh's Medical Center's website titled "This Artificial Sweetener Could Make Cancer Treatment Less Effective." So you can print that, bring it into your doctor, talk to your doctor about it before you make changes in your diet and what supplements you take. So, just a sidebar on that. So, I would like to ask you, you mentioned monk fruit, we mentioned stevia. Do we know, is there a rank order of naughtiness with these sweeteners? you know,
Yeah,
how
so
how bad are they, and what's worse? Does sucralose hit the very top of the list in terms of this seems to be the worst of the worst? How do they compare?
yeah, so at least in the patient samples and those the treatment groups that we looked at, sucralose seemed to be the worst. Um, and it's also one that's consumed quite highly. And then when we looked, saccharin is another very common one that's in pink packets, and that one was not great either. Aspartame Interestingly, we didn't see, it didn't look great, but there wasn't a significant drop in any of the metrics we were reading out. However, there's been a number of other studies unrelated to cancer, some looking at heart disease and things like that. And there's certainly a very strong correlation between aspartame use, those sorts of diseases and things. The other couple that we looked at, so there's also one that's called ACE-K or acesulfame K. This one's interesting because it's often just used in conjunction with another sweetener. So lots of times you'll see sucralose plus ACE-K.
I see.
And so that one really comes down to what other sweetener you're consuming. Whenever we looked at, in our preclinical models and then also in the previous literature, and we're still doing more work on these, with patient cohorts and things like that. But the one that certainly seemed to be the safest was stevia, or kind of these more natural products. I don't think the initial paper looked at monk fruit specifically, but it kind of makes sense. So the ones that were, again, a natural product seem to not have as negative of an impact. Perhaps because, over time we've kind of evolved to, our bodies and microbes have evolved to be able to break those things down. And it makes sense. It's something you would see in nature. With that, though, I always say, stevia to me doesn't taste very good. So, so it's kind of this funny thing, but those seem to be not a problem, at least in what we've looked at so far.
With immunotherapy, specific to immunotherapy in cancer.
Yeah.
So just a question, it's okay if you don't know this, but the public has come to rely on the government to vet products that end up on our shelves. Sucralose is a product that's commonly used. It's been around for a long time. Was there, if this is really not good for our bodies, was there much research or any that went into it going onto the shelves in the first place? Do you know?
Yeah, so this is a question I had actually in the middle of my study. I started thinking how did this happen? How did we end up with something that tastes so delicious and is so terrible for you, right? So I actually went back to some of the original studies that were done. And, what they were looking for at the time was to say, if you, and these were all done, they were rat studies essentially, but basically if you consume this chemical, which is what it is, if you consume this chemical, will it stick around in your body and have any negative effects by that happening? That's the main thing they worry about. Will it hang around? Will it stick in your tissues? That sort of thing. And so that initial study was really just saying, how long is it in your body once you ingest it? And how is it excreted? So is it excreted in the urine, the stool? Is it not well excreted? And so that initial finding, it looked like the vast majority of it was excreted very quickly within a few days. And so that was kind of and of course everybody tolerates it fine. And so that was kind of that initial study saying, okay, this should be okay. There were, I think it was a follow-up study that did actually start to point out, there were some long-term stores of this in the fat. And that wasn't really talked about at the time. But, all these kind of disease-specific things really didn't come about until much later, and I think some of it had to do with people saying, talking to their physicians and then clinical trials coming out where we actually started to look a little bit closer. So none of that was really assessed at the time outside of just saying, it's excreted. It didn't seem to be, and I don't know the literature fully, the carcinogen aspect, but clearly there was something done where they didn't feel like it was an overt carcinogen, but that was kind of it.
Yeah, it's tough, because science evolves. And so we are able to look at things on much lower levels than we were before to really understand how things work that may not have been available when this came onto the market.
Right. And I think, one of those aspects, and this is a really good point, is, when this first came on the market, there was far less being looked at in the context of the gut microbiome, for example. People weren't really thinking about it as much. And so that naturally would not be a readout that people would do. And even now, there are a lot of people who have studied this and done really lovely work on it. But if they don't think about the gut microbiome aspect, then they just don't know to look for it.
Right.
And so what you would hope would happen is that what you said, right? Science evolves. And the beauty of science is that and the way that it works and the reason that it works so well is that we learn something, we put the information out there by publishing, by talking about it, and then it allows, the general public to have that information, but also other scientists. So then they can go and build upon that finding, right? Which is exactly what we did with this, building upon previous literature. And then that way, hopefully we can learn more and better inform, any clinical decisions that might be made on the clinical side, but also informing patients, of what is safe, what's not safe, and that sort of thing.
One of the things I found very interesting is that the impact of eating something and the impact that item has on the gut microbiome
Mhm.
has an impact systemically
Mhm.
within our bodies.
Mhm.
So it didn't matter if it was a lung cancer or a melanoma cancer,
Mhm.
and probably any other cancer. Can you speak to that and how you think about your work with the gut microbiome and immunotherapy? And is it localized to the microbiome? Is it systemic? How do you think about it?
Yeah, so this is actually a really fascinating area of research and one that we think about a lot. And so this is actually this question. There's a question that actually was the basis of me starting my lab completely, which is why would anything that's happening in your gut microbiome matter for what's happening in melanoma or in a brain or in the lung or something like that? Why would that matter, right? They're two completely different tissues, and you do have microbes across a variety of different tissue sites, but it's not like, we're walking around with, you know, they don't have free reign over every tissue site either, right? And so this is a question that we still don't have a full answer to. This will probably take a career to figure out. Hopefully we can figure it out. But what's really interesting is that it's been shown time and time again that, again, shifts to the gut microbiome, whether those are good or bad, has a huge impact on your immune response at other tissue sites. And we think that there's a couple of ways this could be happening. And it's something that we're actively studying now. So the first, which has been looked at quite a bit, but we still don't have a full understanding of it because it's quite complicated, is whether you have what's called bacterial translocation, which just means that you have bacteria in your gut that then move to a different tissue site.
Hmm.
We know that this is possible and can happen. We don't fully understand how it happens and also how that is regulated, right? And what that would do. So we do know that some tumors actually have bacteria in them. And so now we're trying to understand, is that good for the tumor or is it bad for the tumor? And
Do
could you
all
actually
Oh, sorry, do all tumors have bacteria in them or just some?
So we don't know completely. I think, and the field has tried to look at this in a number of different ways. And the field, I think the technologies and the techniques that we can use are finally starting to catch up with the questions. And so I would say, from what has been shown, it seems like the majority of tumors do have some amount of bacteria. And that will vary based on a lot of things. So, how closely related, is there a shared blood flow between the gut and that tissue? Like the liver is a great example of this. Is it a barrier site? So is it in the skin? Is it in the lung where you're already exposed to other microbes outside? But we do know that there is evidence of microbes in most tumors and most metastases, which is very interesting.
So more in metastases than in a primary tumor, maybe, or no?
We don't know actually. I mean, the only thing that has come out on this, which I do find really fascinating and makes it even more complicated, is that we don't know in terms of bacterial load. I think there's some new technologies coming out soon that will actually let us measure that directly, which will be hugely impactful. Um, but what's really interesting is that there have been reports of patients with, with, let's say, with melanoma, metastatic melanoma, and they have mets in different tissue sites, and that each met, whether there's two mets in the liver or a met in the liver and somewhere else, that they seemed to have distinct microbiota in them, right? Meaning that not all, even within the same patient with one type of cancer, the different sites might have different bacteria in them. And what that means, I think, from a research perspective, is one, that this is a very, very complex problem that we need to understand. And two, that perhaps, and something that, that we think about a lot, is perhaps there are shared, maybe the bugs by name, right? We have a million different crazy names for these things, but maybe by name they're different, but maybe by function or the impact that they're having on the tumor maybe is the same. So trying to think about it from that perspective, might help.
It's interesting because I know for some patients, they may have, a tumor in their lung and a tumor someplace else.
Uh-huh.
And perhaps the lung tumor resolves, but the other tumor doesn't. So, one cancer, but you do see different spots reacting differently to the medication. Do you think that is connected back to the bacteria?
It might be, which would be really exciting. I mean, again, it would be a complex problem, but it's an exciting thought, right? Because especially with immunotherapy, the beauty of it historically, and there's a lot of different factors that could be at play, but the beauty of it historically is that, that it does work for a lot of different tumor types and tumors in different tissues and mets in different tissues, because it's activating your immune system, and your immune system is everywhere and it goes everywhere. And so presumably, if you can just boost it in the right way, it should work for everything. And with some patients, that's true, which is absolutely incredible. But for the case where, you have some tumors or mets go away, but not others, I think, the microbiome could certainly be playing a role there, because we do know that there are certain bacteria that are associated with tumors or that live inside tumors that are helping the tumor. And it can do this in a lot of different ways. But part of it can actually be to prevent your immune system from doing its job. There's other things, of course, too, like like a liver versus a bone, like different tissue sites and different organs will certainly have a different environment to start with. So that can also play a role. But certainly, yeah.
You mentioned the liver, and I actually wanted to ask about that. Because I saw something in your eyes where you're like, the liver.
Yes,
So
yes.
tell me, what is it about the liver? Because that is a common location, especially for metastatic breast cancer patients, where cancer often travels to. What is it about the liver that's interesting to you?
Yeah, so the liver, so the first reason that it's interesting to me is that it often is associated with patients who are very sick and where treatments don't work. And that's often, that's where we like to focus. Um, and we do a lot of colorectal cancer work, and that's for them as well. This is a very common place to have mets, and it can be a very, very poor prognostic. But the other thing, and the thing that makes it kind of exciting, both about colon cancer, but also about liver mets from any cancer type, is that the liver, because of where it sits in the body and its relation to the colon and how the blood flow is connected, you will certainly have microbes in the liver. And you can certainly have bugs that have come from the gut that might take up residence in the liver. And so what that means for someone like me who studies this is that we already know that that's happening and that's there. We also know that these liver mets can be really, really just devastating to patients.
Right.
So the hope, and this is, it's like the researcher side of me, the hope is that if you take those two things, right, we know that it's a horrible problem, and we also know that there are bugs there, means that there might be an opportunity for us to start to shift the microbiome within those mets or within the colon and have a direct impact there. So that's why the liver is particularly exciting to me.
Interesting. So if a patient was was struggling with getting a response out of their immunotherapy drugs, and and especially if they had mets to the liver, making the decision to cut out sucralose and improve their gut microbiome through a whole food plant-based diet. That doesn't mean vegetarian or vegan, everyone. That just means like mostly vegetables and a lot of fiber in your diet. Perhaps that could impact not just how their immunotherapy is working, but maybe even those liver metastases.
Hopefully, that's what we're hoping for, right? And I think that there's so many, and again, this is where, we can model a lot of this, and we are in mouse models. And then this is where really having, again, which I've plugged I plugged a number of times that having these great clinical collaborators are huge, right? These really like these physician scientists are like superstars, right? Who can actually do all of these things. So, and what's kind of amazing about this, right? So I think certainly that's a possibility. I hope actually that that is true, because again, that means that that's something that we can change and that patients can change.
Mhm.
But what's great about this too is, to work with physician scientists who are able to, oftentimes make observations from patients, right? From saying, well, I've seen so many patients, and we have another project that actually, is kind of like this, where they say, oh, I keep seeing this with my patients, right? And there's no trial on it yet. There's no study on it, but they see it a lot. And so then when you are able to get, put those people in the same room as researchers, that's where like the magic can happen, because you can say, oh, well, what if it is the microbiome? Have you looked at the mets of those? Have you like, do they have bacteria there? Is this a problem? And then they can say, oh, maybe that's it, right? And so that's where the magic happens. So certainly from a research perspective, I certainly think that's a possibility, and it would be a fantastic thing to be able to look at, because again, as you said, liver mets it's a huge problem, and it happens in a lot of cancer types.
Yeah, it sure does. So let's wrap up talking a little bit about the future for you and your lab. Can you tell us some of the exciting work that you do have on the horizon, any projects in play at the moment for you?
Yes. So we're working on a lot of things. So in the diet space, we're actually trying to take what we have learned from artificial sweeteners and, not only dig in and try and understand more about why these are so bad, but also to broaden our scope and start looking at other dietary factors as well. So this might be things, like, we talk a lot about like a Western diet. It's kind of like the Big Mac American diet, trying to say, okay,
Okay.
how does this impact things? Or an ultra processed foods diet. There's a lot of things, a lot of dietary components that have changed quite a lot over the last, even 20 years, but certainly 20 to 50 years.
Yeah.
And so trying to understand, there's been a lot of changes in cancer incidence, for example, and some of it has gotten better because of better testing. But for example, colorectal cancer, early onset colorectal cancer has actually increased. And so we're trying to understand, could this actually be some sort of dietary or microbiome component?
Mhm.
And then just more broadly, what can patients do, or how can clinicians help patients? Not just thinking about artificial sweeteners alone, but saying, is this actually representative of a bigger dietary change that we're seeing in the population? So we're looking at that. And then the other thing that we're looking at quite a bit, which I was touching on a little bit before, and this is not only how the microbiome impacts tumors, but also how, and this is like where it gets a little complicated, but how the microbiome specific immune response impacts tumors. And so what that means is you, especially in your gut, or any barrier surface where your body encounters different bacteria, you have an immune response to that, to those bacteria. And it's actually kind of in perfect balance where they don't attack the bacteria too much, because that would cause inflammation. That would not be great for us, but also that they keep them in check a little bit. And so there's this whole immune response to bacteria that exists in your body. And normally we have no idea about it. If you have something like IBD, then you do know about it.
IBD is irritable bowel syndrome or
Yeah. Irritable bowel disease or syndrome. Yeah. So unless you have some sort of gut inflammation, we would probably never think about that, right? But what we have found, first in colorectal cancer, and we're now looking at it in melanoma as well, which is very exciting because it is distant to the tumor or distant to the colon, is that the microbiome specific immune response can actually impact immunotherapy response, right? And what we think we can do, what we think we're finding now is that you can kind of fine tune that microbiome response to actually boost immunotherapy response. So rather than saying this is going to be a bad thing, can we kind of rig the system in our favor where those cells can come in and help as well? And so those are a couple of things we're working on. The third thing that we're very passionate about in the lab that's newer, but I think very important, is studying all of these things, diet, microbiome, in the context of adverse events or toxicities. So for patients who go on immunotherapy, and this is becoming extremely common, for patients who go on immunotherapy, they do respond, but they have these, and they're called immune-related adverse events,
Mm.
or bad side effects, also known. But they can be really horrible, right? They can be so bad that oftentimes they have steroid treatment, immunosuppressants, or they have to stop immunotherapy completely, which is heartbreaking because they are actually seeing a benefit from immunotherapy.
Right.
So we're also thinking about those things as well to say, how do we balance this, right? Where patients can not only respond to immunotherapy, but be able to stay on the treatment long term if needed.
That's really exciting. I love all three of those
Thank
areas
you.
seem super exciting and important to patients. Coming back to how, what patients can do in terms of control. I had seen some research suggesting that eating 30 different fruits and vegetables a week
Uh-huh.
would generally provides a healthy gut microbiome.
Uh-huh.
Do you have any, besides the whole food plant based, do you
Uh-huh.
have any more specific guidance for
Mhm.
cancer patients and what you can do and how you can determine if you have a healthy gut microbiome?
Yeah. So I think what you were saying, fruits and vegetables, those are all great things in high fiber. Um, those are all great. This group, so it's the Sonnenberg group out at Stanford. I referenced them earlier, and they've done a lot, not necessarily all in cancer, but really just how do you have a healthy gut microbiome, right? And how does this impact everyday health? So the other thing that they have found is not only things like fiber, but also fermented foods. We now know is really good. And essentially all of that boils down to feeding the right bugs in terms of fiber, and then
Mhm.
also adding diversity to your gut microbiome, and that can happen directly through fermented foods. And so I think those have been the two that have really stood the test of time. The other thing that's being actively researched as well is a Mediterranean diet. And that kind of, when you kind of were describing like a healthy plant-based diet,
Mhm.
the Mediterranean fits in very well there.
Yeah, right.
And so I think it's an added mix of, how do you diversify your gut while also feeding the good ones that are already there.
Mhm.
In terms of knowing if you have a healthy gut or not. So, I think this is tough, and I'm sure there are physicians who could answer this in a much better way than myself. I think the way I think about it anyway, and again, I'm not a physician, but the way I think about it is, a lot of it has to do with how your gut feels, how much energy you have. Because I know if I eat healthy, I have more energy. There is a very direct correlation there. How your gut feels, and then also, the time to do it. Is it normal? Is it not normal? And GI docs are great about being able to kind of help with this and say, is that normal? Is it not normal? And you can actually learn a lot. We always have like working in a lab on the microbiome. You can imagine all the funny little things that we end up saying around the lab,
Yeah,
about poop and others. But really, that's the best readout we have. You know,
right.
and so I think thinking about it from that perspective, just being cognizant of what you're putting into your body, what you're eating, and then it's really, what is it like on the other side? And GI docs are very good about that, and kind of talking you through that. And it's something that it is funny because, I always say like, there's like jokes around poop and everything, but it is something that people often don't want to talk about,
Right.
but it can be a really important warning sign that there's something wrong. And so I think it's something that, more people should actually be talking about, and not having it be such a thing you don't want
A
to
taboo.
a
Yeah.
taboo topic, yeah.
I've learned over time, it's like you have messy disease and I've had two C-sections, I've had bilateral mastectomy, I've had all sorts of stuff. And at the end of the day, talking about poop is hard, is a lot less hard than it was years ago before it
Yes, I'm sure.
happened.
Yes, exactly, exactly.
And I will say I had a lot of disruption in my gut microbiome. I had a lot of diarrhea. And I did a test called the Genova
Mhm,
diagnostics test, which is a comprehensive test, and it does break down for you a lot of different
mhm,
bacteria, fatty acids, long chain fatty acids, short chain fatty acids.
mhm,
And, listeners out there might say, why do I need to know that kind of stuff, that detail? But it was helpful as more information comes out about what bacteria are good to have, good bacteria, what bacteria are bad bacteria,
Yeah,
and where you might have overgrowth or undergrowth. It can be a roadmap if you're working with the right doctor. And there
absolutely.
are doctors out there that are excellent in being able to take that information and do something for you that will change your gut microbiome, that will help you.
Yeah.
I was able to get a series of supplements, and I thought, oh gosh, I'm going to have to take all these supplements forever. But the doctor was like, oh no, take
Yeah.
them for 2 weeks and you'll feel much better. And sure enough,
Yeah.
I did. And I thought, because I'm still on therapy, I'm still taking drugs because I'm metastatic. So I get my treatment every 3 weeks. I thought, well, I was just destined to have a terrible gut microbiome. But I want to tell patients out there, don't think because you're in treatment that there's nothing you can do. It may be
Absolutely.
harsh on your gut, but there are things you can do. And taking a test like the Genova test, there are other brands, and working with a doctor who specializes in the gut microbiome can help you with relieving symptoms and making it healthier. And that health may impact your
Yes.
survival.
Absolutely. 100%. Yes. And I think so. One other thing, which I didn't bring up, but I'm sure that Dr. Schmitz, when she was on, brought up, which is exercise. And exercise can actually have a positive impact on your gut microbiome as well, which
Yes.
is really interesting. It's a whole other area of research. And I think, what you said about this test is, I'm so glad that these things exist and that physicians are starting to, really pay attention to this. Again, this is my area of study, but I would love it if this became more of a normal thing, kind of like, blood work, even if you're healthy or even if you are sick, and saying, like, let's just see, like, how are things looking, right? Are your lymphocytes normal? Are your white blood cells normal? Also, like, is your stool normal? Your gut microbes normal? And, it makes a huge difference in the way you feel, but it can also have a huge impact on how well you do, right?
Right. And your
Yeah.
survival. I mean, that's
Yes.
what sometimes we hate to say those things because nobody wants to think about dying. But
Yeah.
this is cancer and it's so real. And if we get real about it and we say these things out loud, then we can do something about it. And the
Absolutely.
important thing is, this has an impact on progression free survival that we know we have data about. And we're looking to see what will happen with overall survival, but
Yeah.
it's looking like it may impact overall survival as well. So we really want listeners to lean in and think about this, because you can make a change. You can have control over this. This is something you can do for yourself.
Right. And I think, the one other thing that I will say too, and this is something we're actually studying, is you brought up this idea of, all of these treatments that people are getting. It can be hard on your gut microbiome. And so we're actually looking at this now to try and put some causation and, strong correlations there, just to give patients, hopefully more opportunities to restore their gut microbiome, that, as they go about their treatment, because there are so many treatments that can negatively impact it. And so I think, this idea of, doing things yourself and getting those tests and readouts is amazing. And hopefully, as these preclinical trials come out and clinical trials, hopefully we can use this again as, like, you get blood draws so often to make sure you're healthy. Let's also check your microbiome, make sure that's healthy, right? So
Right.
hopefully we can get to that point. It would be great.
Awesome. Awesome. Well, gosh, we've learned so much from you today. Thank you so much for coming, Dr. Abby Overacre. You have been really helpful, and we're so grateful for all the research you do.
Thank you so much.
All the information that was provided today was for educational purposes only. I'm a patient, just like you. I am not a licensed or accredited physician, therapist, or clinical researcher. All information provided is not intended as a substitute for the advice of a physician, therapist, nutritionist, or any other qualified healthcare professional.